Today we will see paper 3, B.Sc paper first, Animal Systematic and Diversity 5th. Myself Mrs. Adhav UK, Department of Zoology, रयत शिक्षण संस्थाज आर.बी. नारायणराव बोरावके महाविद्यालय श्रीरामपूर, District Ahmednagar. Today's our topic of discussion is Protozoa, general topic Protozoa, from which we will see locomotion in Protozoa. What is Protozoa? First we will see Protozoa are microscopic unicellular organisms, or they are the eukaryote organisms. These microscopic organisms body is formed with a single cell, and these protozoan animals found worldwide, or they found throughout the world and with variable habitat, that is they found in most habitat. Some of them are free living, some of them are parasitic. These protozoan animals which are parasitic or free living, they shows complex internal structure with complex metabolic activities. Their size is from 1 to 150 micrometer in diameter. They encloses their body with plasma membrane, and in that plasma membrane the cytoplasm is present, which shows two parts, outer ectoderm and inner endoderm. Peripheral is the ectoderm and central is the endoderm. From the ectoplasm, locomotor organelles arises outside as in the form of cilia or flagella, and in the ectoplasm some locomotor organelles are present inside it, like pseudopodia. These are the pseudopodia. Means there are 4 types of locomotor organelles which are embedded within the plasma membrane. In some protozoans, pellicle is present, which is outer covering. It is constant covering, and this pellicle gives constant or permanent shape, which is rigid, and it gives permanent shape to the some protozoan animals, some of them having plasma membrane, some of them having pellicle. Here today's our topic of discussion, we will start with locomotor organelles, that is pseudopodia, flagella, cilia and the myonems or pellicular contractile structures. Thus there are 4 locomotor organelles present in protozoa. According to their shape, size, structure, they are classified and given here with the diagrams. First we will see pseudopodia. It is also called as amoeboid movement. It is also called as amoeboid movement. This amoeboid movement is exhibited by pseudopodia, and pseudopodia are formed by the streaming flow of cytoplasm. By the streaming flow of cytoplasm, like this. These are not true locomotor organelles. These are false fit. False fit means they are temporary locomotor organelles of some protozoans. According to their shape, size, these pseudopodia or amoeboid locomotor organelles again classified or divided into 4 types. First, the lobopodia, second, philopodia, third, exopodia, and fourth, reticulopodia. We will see first lobopodia. These lobopodia are found in amoeba and entamoeba. These lobopodia are formed by the streaming flow or pressure flow of endoplasm or ectoplasm. This lobopodia, lobe means lobe like, podia means foot, locomotor organ foot. These are lobe-like locomotor organelles. They having rounded ends or blunt end. They are not tapering. They are having rounded or blunt end, lobe-like end, and they are formed by the streaming flow of ectoplasm or endoplasm or pressure flow of ectoplasm or endoplasm. While animal wants to walk or locomote or move from one place to another place, at that time the pseudopodia is formed in front direction, and the opposite direction pseudopodia will disappear gradually or slowly. Means it is temporary locomotor organelles or false feet of the animal. Example shown here, amoeba. These are the lobe-like pseudopodia which found in amoeba, and these are due to the pressure flow of cytoplasm, that is the ecto and endoplasm. Then second type of locomotor organelles or pseudopodia, second type of pseudopodia are called as philopodia. Example of philopodia is euglypha. Here this euglypha shows pseudopodia like the filament. Here small filament which is having pointed end at the tip. From the basal to tip they are filament like or fine thread like, hence they are called as philopodia. Its name again employed by this filament like structure. And these pseudopodia, filament like pseudopodia or philopodia, are formed only due to the pressure of ectoplasm. Example euglypha. Third pseudopodia is called as exopodia. These exopodia are found in actinophyres. These exopodia are elongated compared to philopodia. They are elongated and stiff and pointed at the tip. They are longer or elongated. They are stiff like, and they arises from the axial rod like central part which is surrounded by cytoplasm. These are needle like or pointed like, and there is the flow of ectoplasm and endoplasm in reverse direction or both the direction. Means cytoplasm move from tip to base and base to tip. These are the exopodia, arises from axial rod of the actinophyres. Then fourth type of pseudopodia are called as reticulopodia. These reticulopodia type of pseudopodia found in example Elphidium. Here these reticulopodia, they are having branching like structures, or they forms network like structure. They forms the network like structure. They are elongated and shows two-way flow of cytoplasm from tip to bottom and from bottom to tip. Means there is two-way flow of cytoplasm. Thus there are four types of pseudopodia found in these examples of protozoa. This is the first locomotor organelles found in these animals, and they are called as amoeboid movement. When animal wants to move for some period, then the pseudopodia supports the substratum or any place, and there is the adhesive secretion first, and then the pseudopodia is formed due to the pressure flow of cytoplasm, and then the body is lifted on that direction and animal move forward and forward. This is the locomotor organ pseudopodia. Then second type of locomotor organelles found in protozoa is called as flagella. Flagella is elongated. One or two flagella may be found in some animals. Example of flagella is Euglena and Trypanosoma. Flagellum is single, elongated or large. Sometimes it arises from front side of the body and sometimes it may arises from the ventral side of the body. Flagellum is typical structure which consists of axoneme. It consists of axoneme which arises from basal granule of the kinetosome or blepharoblast, and in the axoneme there is 9+2 arrangement. 9+2 arrangement means towards the periphery there are 9 double fibrils are present like this. 9 double fibrils are present with 2 arms, A and B arms. Like this, there are 9 double fibrils with 2 central fibrils. This is the axoneme having 9+2 arrangement. There are 5 different types of flagella found in protozoan animals. Flagellum shows, suppose this is a single flagellum, it shows small hair-like structure. Small hair-like structure present on the flagellum. This small hair-like structure is called as mastigonemes. And according to form structure, they are classified or they are divided into 5 categories due to the presence of mastigonemes or hair-like processes present on them. First is called as anematic or acronematic. Example, Noctiluca. When there is no mastigonemes, single flagellum is present without mastigonemes or mastigonemes are absent, then it is called as A means absence, anematic type of flagella. Second type of flagella is called as stichonematic, which is having on one side mastigonemes, only one side mastigonemes present throughout the flagellum. Example, Euglena. Then this type of flagellum is called as stichonematic, one side row or one row of mastigonemes. Third type is called as pantonematic. Example, Paranema. From the basal body, 2 or more rows of mastigonemes or hair-like projections present from tip to the base. These are called as pantonematic, that is both right side and left side of the flagellum shows mastigonemes or hair-like projections, pantonematic. Fourth is pentachronematic. Here Erosilus example shows the flagellum with 2 or more rows of mastigonemes, but anterior side there is no flagellum. This part of flagellum is called as terminal filament. It is called as pentachronematic. Example, Erosilus. Then the fifth one, acronematic. Example, Polytoma. Here the entire flagellum is thick. Entire flagellum is thick up to 1/3 part of the body, but the distal part shows the terminal filament or it is thin, which is called as terminal filament. These are the types of flagella, and the movement or locomotion in those animals which shows example Euglena, Trypanosoma or flagellate animals, those shows this type of flagellum which helps for locomotion in protozoa. They are called as flagellar locomotion, or the organelles which help for locomotion with the help of flagella. They are called as flagellar locomotion. This is the second type of locomotor organelles present in protozoa. Third, the cilia. Cilia are numerous, but they are short. As suppose this is the flagella. What is the difference between flagella and cilia? Cilia are small and short, but numerous. They are numerous and covers entire body of the animal. Example, paramecium. But in case of Euglena or Trypanosoma, flagellum is single and elongated or longer. They are longer. Here, mastigoniums are absent in cilia, but here mastigoniums may be present on one row, one side, or on both the sides, mastigoniums present on the flagella. Then, flagella shows locomotion in forward direction or backward direction, but cilia move in coordinate manner. In coordinate manner means, suppose there are 3000 cilia. Their number is numerous in paramecium, but suppose we consider here 300 cilia may be present on the external surface which arises from the pellicle. They all move in coordinate manner, means one after another, and there is contractile movement from base to tip. First there is effective stroke and then there is recovery stroke, like this. They move one after another, 1, 2, 3, 4, like this. They move in coordinate manner, but here flagella doesn't show this type of locomotion. This is the this type of locomotion which is found in cilia or sporozoa. They are called as ciliary locomotion. And fourth type is pellicular contractile structure or the myoniums. They are found in ciliates and flagellates, also in sporozoans. These pellicular contractile structures or myoniums are the ectoplasm. They are present inside the ectoplasm. They are present inside the ectoplasm. They are in the form of large branches. Sometimes there are single branch, sometimes there may be a network of branches in the myoniums. These myoniums shows internally canals or ridges or grooves, and on that grooves or canals or ridges there is the presence of light and dark substances. Example, present in stentor. And this type of locomotion which is effected or exhibited by myoniums is called as pellicular contractile structures. And thus there are 4 types of locomotor organelles present in protozoan animals. That is pseudopodia. It is also called as amoeboid movement. It is found in amoeba and other animals. Flagella, it is found in Euglena, Trypanosoma, etc. Cilia exhibited by paramecium and other ciliate animals, and pellicular contractile structures or myoniums present in ciliates, flagellates and sporozoans. We will stop here and see next topic in next video. Okay.